morning, everyone. Thank you, Brenda, for the very kind introduction. I'm going to go ahead and get started. As Brenda mentioned, today I'll be talking about bioequivalence standards for both generic topical and transdermal drug products and focusing on what we have been doing as it relates to innovation. You heard Dr. Rane talk about all the research we have been doing, and I'll talk a little bit about how we are incorporating the research into our guidances, and then harmonization. The goal to provide consistency as well as clarity related to the regulatory expectations that we typically lay out in our guidances. Before I get started, my disclaimer, and then a brief outline of my presentation where I'll first focus on the TDS, the general guidances and the product-specific guidances, and then the topical drug products or topical dermatological drug products where I'll specifically be speaking about the product-specific guidances. So beginning with the transdermal drug products, what I have on the slide here are the two most commonly known designs for transdermal delivery systems, which may be referred to as patches or extended release films elsewhere. And these are the reservoir TDS on the top here and the matrix TDS on the bottom of the slide. The reservoir TDS usually has a liquid-filled reservoir. There may be unique failure modes associated with these products, and therefore, if you are considering developing a generic product with a reservoir design, we recommend that you look at the guidance that is noted at the bottom of the slide, the refuse to receive guidance, and talk to the Office of Pharmaceutical Quality as early as possible during your product development program to discuss the viability of your product design. The matrix transdermal systems are more widely used for transdermal products that are currently available in the market. The most simplistic design, you have the backing membrane. This is the part that you can see when the product is applied on a patient. The drug adhesive layer, which is responsible for delivering the drug. And then you have the release liner, which is removed prior to application of the product on the skin. And like I said, this is the most simplistic design of a matrix-based system. There are design variations that are available, such as in the shape of the product, in the shape of the product, in the size of the product, as well as in the components and the composition that are included in the product. And the important thing for us to note for the purposes of this presentation is these are allowable differences between a generic product and a reference product as long as bioequivalence can be established between those two products. However, these differences also mean that there are different failure modes that are related to product design, which needs to be mitigated during the product development plan. And therefore, we recommend, to evaluate, say, for example, potential differences in adhesion between a test product and a reference product that may arise due to differences in size, shape, or composition, we recommend an in vivo adhesion study to evaluate differences in the sensitization potential. If there are any differences in the sensitization potential between test product and a reference product, we recommend an in vivo irritation study. And I'll be talking more about the in vivo studies that we recommend typically for establishing BE for transdermal systems. Dr. Rotten, during his talk, will be talking about some of the quality aspects that needs to be incorporated within the product development plan that may arise due to differences in the product design. So for the in vivo studies, for a typical transdermal product, we recommend a BE study with pharmacokinetic endpoints, and we have a guidance for that, a general guidance. This is the same guidance that is used for solid oral drug products for conducting PK studies. So I'm not going to go into a lot of details related to the design and conduct of the pharmacokinetic studies. As I said, we recommend an in vivo comparative adhesion study. We have a general guidance for that. And we also recommend an in vivo comparative irritation and sensitization study. And again, a general guidance for that as well. And I'm going to talk about each of these two guidances on the subsequent slides. So beginning with the general guidance on adhesion, this guidance was first published in, the draft guidance was first published in June of 2016. And prior to the publication of this guidance, we did have recommendations for design and conduct of adhesion studies, but they were available across about 20 product-specific guidances. So as Dr. Rane mentioned, we consolidated our thinking related to study design considerations within the draft guidance and provided generalized recommendations that are applicable to all transdermal products within the first draft version of this general guidance. We also introduced a new statistical approach. Again, the goal here was to ensure that powerful statistical approach is available to the industry to ensure that high-performing generic products can get onto the market faster and made available to the American public. We also provided some critical information related to study control, such as tampering with the TDS system is not allowed during the study, or normal subject motion should not be restricted, such as subjects should be allowed to shower during, say, for example, evaluation of a 7-day transdermal product, and so on and so forth. However, and we think that this guidance was hugely successful in that we were able to use the statistical approach to get a lot of high-performing generic drug products onto the market. However, as with any guidance, we had a feedback period, and following feedback from internal as well as external stakeholders, we published a revised guidance in October 2018, which provided additional clarity related to how data should be collected and analyzed during the addition studies, as well as the potential use of alternative scales in addition to the 5-point scale that we typically recommend in our guidances. In conjunction with the publication of the revised addition guidance in 2018, we also published a new draft guidance for assessing irritation and sensitization studies for a generic TDS product in October 2018 as well. And in this situation, a very similar concept, we consolidated thinking that was at that point present across 24 different product-specific guidances, providing generalized recommendations related to study design and conduct for irritation and sensitization studies. Again, we introduced a new statistical approach, which we think is a more powerful approach to enable, to help move more generic, high-performing products onto the market faster. A new concept was introduced related to where sensitization studies may not be appropriate, such as when you know that the active pharmaceutical ingredient itself is sensitizing. And then study design controls similar to what we had discussed for the addition study, but controls which are more relevant to the irritation and sensitization study. Once we had all these three guidances, so the guidance on design and conduct of the PK studies, which has been available since 2013, the addition guidance as well as the irritation and sensitization guidance, as soon as these three general guidances are available, in October 2018, we also published 27 either new or revised PSGs for most of the transdermal delivery systems that were on the market at the time. And the goal here is to streamline our recommendations and provide only product-specific information related to each of the studies. The PK study, the addition study, and the skin irritation sensitization study, information such as the design of the study, the strength of the product that should be used in each of the studies, and the subject population were retained within the product-specific guidances, but all general information that are generally applicable across the studies are now available within the general guidances, with the idea that we should be able to use the general guidances in conjunction with the product-specific guidance to develop the overall study protocol. We did offer some clarity related to different aspects of these three studies within both the product-specific guidances as well as the general guidances that I would like to take this opportunity to highlight today. For the next 7 slides or so, these slides are a little wordy. They are directly obtained from PSGs, and I have hyperlinked them to the PSGs that the text is from. So I would request if you can focus on the sections that are underlined, I will be really grateful. So the first clarification that I would like to highlight is related to integration of PSGs with external references. What do we mean here? We mean that in general, when a study protocol is being developed, say, for example, inclusion-exclusion criteria for recruiting subjects, healthy subjects into a PK study, information about where the product should be applied on the body can generally be obtained from the product label. We still retain the information related to the duration of the study in the product-specific guidance, but the expectation here, again, is that we are able to use the product label in conjunction with the guidances that we have available to develop the overall study protocol. The second clarification is related to data connection and analysis of the data that is for the PK study. The clarification here is we observed a trend where in the past, protocols had criteria where if the addition score reached, say, for example, a value of 2 for a specific subject in a specific arm of the study, the sampling protocol or the blood samples that were being obtained from the subject were immediately suspended. Our recommendations currently indicate that please continue to obtain the PK samples and analyze the data for all samples, because this gives us the opportunity to understand how the degree of detachment, how as your transdermal system begins to come off, if it does, how that impacts bioavailability, and that helps us to make a better determination of the comparison of a test and reference product. We also provide some clarification related to there should not be any deliberate action to reapply the product during the scope of the study, or overlays and tape and such should be avoided throughout the study. The third clarification that we provide that I would like to highlight today is related to establishing proportionality. And this example is from a reversing mean transdermal system. So the strength of the product that is evaluated in the BE study in this case is the 9.5 milligram per 24-hour strength. And typically, when there are multiple strengths of a transdermal product that are available on the market, you can request a waiver of in vivo testing for the alternate strengths, such as the 4.6 milligram and the 13.3 milligram strength here, as long as there is an acceptable BE study, an acceptable in vitro dissolution or release study release results available, as well as the TDS formulations are proportionally similar. There has been some confusion as to what proportional similarity really means for a transdermal system, and therefore we provided some clarity in the guidance, but I'm going to take the next slide to explain what we really mean here. So again, with the reversing mean system, you have the reference strength which is evaluated in the BE study, and you can see that if you look at the smallest strength, the reference strength is twice the size of the smallest strength. But if you look at the nominal value of nominal strength that is reported for this product, 2 times 4.6 is not 9.5. So when you design your reference product, we encourage you to look at the ratio between the different strengths of the active surface, the ratio of the active surface area between the different strengths of the reference products, and design your generic product accordingly. So again, proportionality should be based on the ratio of the active surface area among the different strengths of the product, and not on the nominal strength when these two values are different. The last clarification is related to the dissolution studies. We have had some language in the past related to multiple pH, etc. in the past. We have now simplified that language, and currently our recommendations indicate that you have to carry out the dissolution or release studies using 12 dosage units for each strength of the product. And then for developing a method, you may be able to use the method that is typically available within the dissolution database as a starting point for developing a discriminatory method for comparing your test product to the reference product. Moving on now to the addition study. Again, our main goal was to make sure that there is alignment between all the general and the product-specific guidance. And as I mentioned before, in our product-specific guidances, we do highlight that you conduct a PK study, addition study, and the irritation and sensitization study. But one clarification that we offer in all guidances is you may be able to combine the two studies as long as you clearly pre-specify in your protocol how you are proposing to combine the study. Example, if you have 60 subjects for your addition study and you choose to evaluate 36 of those subjects for your PK subpopulation, you need to clearly identify those 36 subjects in the protocol. And then a second level of clarification, if say for example, you decide that you are only going to include subjects who have a mean addition score of greater than 2 for an assessment of the pharmacokinetic endpoints, that is the assessment of Cmax and AUC for comparing your test and reference product. This also needs to be pre-specified in the protocol. So you need to clearly identify your per protocol population for your assessment of your PK endpoints within the protocol. And just a reminder, irrespective of this pre-specification, you should still continue to sample and analyze the blood samples at all time points, so we have the data to correlate the addition of your specific product with the bioavailability of the product. Last clarification related to the product-specific guidances, the irritation and sensitization study. Here we do provide recommendations related to the size of the product that should be used. Typically, it depends on the availability of the different strengths of the product and what is the smallest strength that can be evaluated in irritation and sensitization study. I would like to highlight, however, that please pay attention to the duration of wear for each product or the frequency at which these products should be replaced during the scope of the irritation and sensitization study, because in very few instances, this frequency of replacement of these products over the 21-day period is different from the duration of wear on the product label. So use the product label in conjunction with the product-specific guidance and the general guidances to develop your overall study protocol. The last clarification is related to residual drug. We do have general guidance on residual drug since 2011, and the clarification that I offer here is from the edition guidance, which indicates that for evaluating residual drug, please use the TDS units that have been used either in the pivotal PKBE study or the combined in vivo edition and PKBE study. So with these clarifications and the 27 guidances that we now have published, we hope that we were able to provide consistent structure and recommendations across all PhDs, clarity related to strength, size, duration of the studies, clarity related to the waiver of in vivo testing, removal of studies when they are not needed, say for example a sensitization study when you know that the API is sensitizing, and all repetitive information was migrated to the general guidance with the idea of harmonizing the recommendations with the PhDs, providing streamlined recommendations within the PhDs, just the product-specific components, and then all the rest of the information within the general guidances, with the ultimate goal of course, increasing the overall efficiency of the TDS development programs. So now that we have talked a little bit about the transdermal or TDS guidances, I'm going to switch gears and talk about the topical products. As Dr. Rane mentioned, historically, potential ways for establishing BE for these products were mostly clinical BE studies. And over the last 5 years or so, we have focused more on what we think are more efficient characterization-based approaches for establishing BE, which typically include in vitro-based studies and may or may not include in vivo PK study. I'm not going to go through this slide in a lot of detail because Dr. Rane already talked about what these different modules are: the formulation sameness, the physical and structural similarity, IVRT, IVPD, and PK, except for saying that this is a scalable framework because as the complexity of your drug product increases, so do the potential risks, so do the risks associated with potential failure modes for establishing BE. And as such, you may be able to add additional modules in an effort to develop an efficient bioequivalence strategy, which would mitigate all those risks. So I'm going to go through each of these components as they appear in our guidances and try to explain what the expectation is usually for the recommendations that we provide. The first component is, of course, formulation sameness, and we heard a lot about what our current standards are. This is the guidance that was published last week for metronidazole topical gel, 0.75%, and our current standard indicates, again, no difference in inactive ingredients or other aspects of the formulation that may significantly impact local or systemic bioavailability. So differences in the nominal amount of the pH modifier may be an allowable difference as long as the final pH is matched, but there should not be any differences between the test and the reference product that may impact local or systemic bioavailability when you are using a characterization-based approach for evaluating BE. The second component, Q3. So for a single-phase system, we typically recommend looking at appearance and looking at microscopy. What do we mean? For appearance, we are trying to understand if the look and the feel of the test product is comparable to the reference. With microscopy, we are trying to understand if the microstructure of the product or say, for example, if there are particulate matters in the test which are comparable to the reference. If there are particulate matters, we look at particle size characterization. If there are known polymorphic forms for your API, we look at if the polymorphic forms are matched between the test and the reference product. And the goal here is to ensure that we know that some of these components can impact bioavailability, and the goal is to ensure that there are no differences in the, between the test and the reference product, which could in fact impact bioavailability. We look at drying rate, again trying to understand the metamorphosis of the product over time, because differences in the manufacturing process could impact metamorphosis, which in turn again could impact bioavailability. We try to look at specific gravity, rheology, pH, again in an effort to match all the critical Q3 characteristics in an effort to ensure that the bioavailability from the test product would be similar to that of the reference. For more complex systems, such as multiphasic systems, we are of course interested in all the components that I talked about already on the previous slide, but we are also interested in understanding water activity, specifically for when formulations have less than 50% water, because differences in manufacturing process could lead to differences in that encapsulation of the water in your formulation, which in turn could lead to differences in the metamorphosis of the formulation. We try to look at globule size distribution where it's relevant, and also the impact of the container closure system. And while I'm not going to go through all of this data, microscopy data, spectroscopy data that is shown in the slide here, the take-home message here is when this is a product that is approved in the UK, was dispensed using a tube compared to the pump, there were differences in bioavailability as observed in this in vitro data set just because of how the product was dispensed. What does that mean for us? That means when a product is being designed to be packaged in two different container closure systems, you may need to evaluate the dispensed products from both the container closure systems and ensure that bioavailability is not impacted due to the dispensing mechanism. Moving on now to IVRT. We have recommendations related to IVRT laid out within the acyclovir cream guidance, which is hyperlinked here. I don't have a lot of time to go into all the details, but what I would like to highlight here is the three components. There are three critical components of any IVRT method that is used to support an evaluation of BE. The first one being the method development. Here the expectation is you will look at the receptor solution, the type of membrane that is supposed to, that can be used for your particular setup, the apparatus, the dose, the duration, and once you have finalized these components, you take that method into validation and look at specificity, selectivity, and sensitivity of your method. Once you have the validated method, you then use that method to compare the test product to the reference in the pivotal study. And an additional clarification here is if you have product that is packaged within different container closure systems, again, you may need to conduct IVRT studies with products dispensed within with each package from each of the container closure systems. For IVPD, again, a similar concept. We have the recommendations laid out within the acyclovir cream guidance. Again, highlighting that conceptually there are four unique steps for IVPD method. First one, method development. Again, you are figuring out what type of skin to use, what type of barrier integrity test, the apparatus, the dose, the duration. Then you take that method into validation where you are looking at sensitivity and selectivity. Once you have a validated method, you run your pilot study with the components that you already have from your validated method to understand the variability that you expect to see in your pivotal studies. That gives you, pilot study helps you to understand what variability you expect to see across different donors for your reference product, and then you can use that data to power your pivotal study adequately. One quick clarification here, please lay out your method development and validation strategies clearly within the study protocols as well as your statistical analysis plan, and this is very important for our IVPD study. I'm not going to talk about the PK studies because they are well understood, but if you have any questions related to any of these last three studies, please feel free to bring them up during the discussion session. In summary, I would like to say that when a PSG is available for a topical dermatological product, you may choose to follow the recommendations within the PSG or talk to us through the pre-NDA or controls correspondence process. If a PSG is not available, identify your reference product, and then you can develop a product development strategy based on what we have talked about, how we go about identifying potential failure modes for BE for the products and developing product-specific guidances. Similar studies may be used to develop your overall product development strategy. With that, my acknowledgements for our entire team within the Office of Research and Standards and the guidance development teams that are spread across the Office of Generic Drugs as well as the Office of Pharmaceutical Quality. Thank you.